Awesome. All right, welcome everyone to our seminar. We're super happy to have Professor Serge Belongie with us today, giving a talk on training machines to identify species using GBIF mediated data sets. I'm very interested to find out more about this. I wanted to give a brief introduction. So Professor Belongie is a professor at Cornell University and received his PhD from Berkeley. And he's been doing lots of great research at the intersection of computer vision, machine learning, crowdsourcing, and some research with birds, as you can see. He's also co-founded many companies, received an NSF Career Award, the Alfred P. Sloan Research Fellowship, lots of other awards. And so we're super happy to have him here. Welcome, Serge, to our group, and I'll hand it over to you.
Great, thank you. Is the audio working?
Yes.
Excellent. All right, thanks so much for the invitation, Kate and Margaret. If you're online, thank you also. So as Kate mentioned, I have done some work with birds, so that's why we see the picture of the bird there. And I'm a professor at Cornell University, but also I'm an associate dean at Cornell Tech, which is the new, I guess not so new, but maybe 7-year-old campus of Cornell University, which is located in Manhattan. And Kate already went over the research interests. So the title of the talk emphasizes this portion about GBIF mediated data sets, which has to do with connecting the computer vision and machine learning community with the biodiversity community. But this is an offshoot of my group's many years of work in a project called Visipedia. So I'm going to start with some overview of Visipedia, and then talk about a less supervised version of the work we're doing in Visipedia called SNAC. And then that will bring us to the portion that is focused on biodiversity. So starting with the Visipedia part, I think there's still someone unmuted. Okay, so the Visipedia part is focused on something called fine-grained visual categories. And these, we want to recognize these kinds of categories with humans in the loop. So in the Visipedia project, we focus on this kind of specialized knowledge that is held by specific communities, human communities, that know about things like birds or make and model of cars or fashion, apparel, plant disease. So there are communities that focus on these different kinds of problems. But even if you're not a member of one of these communities, you can still appreciate it just from a visual perception point of view. You can see the different types of bills that a bird can have. And you may not know that specialized terminology, but you know it when you see it. And so with the Visipedia project, we want to make that type of content shareable. We want to capture and share visual expertise, even if you don't know that specialized terminology or know about the taxonomies behind that kind of data. So one of the things we're tapping into is the strong interest within hobbyist communities, not just for birds, but there can be different types of animals where groups of people organize on the internet that are fans of that type of animal or a type of airplane. And so groups get together, collect images. They enjoy discussing challenging cases or trying to understand what makes one image a member of one category versus another. So we want to tap into that expertise, learn from those communities, and give back something to them to help people search through or retrieve images that are relevant to those interests. One of the key ideas here is the complementarity between human and machine ability on these types of tasks. So if you look at the image on the left, this question of what basic level category is shown in the image is quite easy. Is it a chair? No. Is it a bicycle? No. Is it an airplane? Yes. So this is basically very easy for humans. The image in the middle, if I just take a person off the street and say, does this image show an indigo bunting or a blue grosbeak, then that is challenging in general if you're not an expert in birds. But if we go to image C and the task shown on the right, and we break it down sort of like a field guide where we say, we're going to ask questions about parts and attributes of this object. Is the belly yellow? Or a part click question like click on the tip of the bill or the end of the wingtip. If you can break down the task into these simple part and attribute questions, then the task becomes easier. So that led us to one of the early approaches that we proposed for Visipedia, which was called Visual 20 Questions. So the idea with Visual 20 Questions is that the image comes in to a block that we've labeled computer vision. So, and this took place right before the deep learning revolution happened. But the idea was that computer vision block encompasses the state of the art in object recognition in computer vision and machine learning. So in an ideal world, that image would come into this block and you would get out some probability of the class given the input image. And you would have a really strong spike around the correct species, indicating a very high likelihood. And then the other species would be very low. But partly because the deep learning revolution hadn't happened, we anticipated that there would be substantial confusion, or at most what the computer vision block would do was say that certain species were more likely than others. And then we would use that to kick off a 20 questions game. So based on what comes out of the computer vision block, we would select a question according to this algorithm, and we would choose out of a set of predetermined questions that question that would maximize the expected information gain. So in this case, based on the short list of species that could match this, we ask a question like, is the belly black? And the answer is no. We update these posterior probabilities. We select another question, is the bill hooked? And typically we found that it required about 2 of these kinds of questions, and then we would narrow it down. So again, this was based on the state of the art at the time. So this brings us to a little historical comment that some of the older researchers among us will remember. So I just use this term antediluvian to refer to techniques from computer vision before the flood of deep learning papers. So it's just a play on the word antediluvian, which means before the flood. So most of you who are students probably aren't citing papers from before 2013, but there were papers back then that talked about manually engineered features, things like histograms of vector quantized filter responses, color histograms, shape context, SIFT. So we used a lot of those techniques, and needless to say, they were basically swept away in their entirety after the deep learning revolution. And so within my group, I still kind of held on to this idea that it would still be necessary to localize parts of birds. My thinking was just, sure, convnets are powerful, but birds are really flexible objects. And I thought it would still be necessary to train a part detector for the head, the body, and then have an entire image representation. And the idea was that we would do some kind of rudimentary alignment and then run those through individual convnets, get some kind of aggregated representation and classify that. And there actually was a period around 2014 where that was the leading approach, but very quickly that too got swept away just by virtue of using even more data. No sort of part localization or alignment was necessary. And I think any of you who are PhD students at this point probably entered the field when this kind of paradigm was already established. So what changed at that point was that it was very, in terms of classic supervised multi-class classification, it became less a game of manual feature engineering and more about curating really high quality data sets. So finding the groups or the organizations that would have the ability to put together really high quality data. And in our case, this is what brought me to work with the Cornell Lab of Ornithology and gave rise to one of these very large data sets, one of the early data sets for fine-grained recognition. So working with them, we first built a web-based interface where you could upload images, indicate geolocalization, indicate when you saw the bird, which taps into this so-called eBird database that has priors for all of these different birds, based on decades of observational data for birds. So the web interface was set up about 5 years ago. And so you could upload an image. In this case, the short list was of length 1. We still had that visual 20 questions thing in there, but as we got more and more data and trained deeper networks, that short list was rarely longer than 2. So this didn't actually require having to click on anything. After observing this and sort of inspecting the thumbnails, you could click, "This is my bird." It would play the bird song, and it would display additional information about that species, at which point it puts it into a bucket, and then someone with sufficient expertise in that network of the Cornell Lab of Ornithology, they can look at it, make sure it's correct, and then, for example, on a monthly basis, the network would be retrained and then get a little bit better for the next batch of users. So this led to the creation of Merlin Bird Photo ID. So this is a free app that you can download for iOS or Android. And this, as you can see in the about information, it's a contribution of the Visipedia project, which is a collaboration between Cornell Tech and Pietro Perona's lab at Caltech. So this is available now, and it caters to an audience that wants on-device recognition. So this is using TensorFlow Lite, and it's based on bird packs. So you may go on a trip where you're not going to have cellular data or Wi-Fi. You go to a region, you download a pack, it might be 100 megabytes or so for, say, Scandinavia, and you just have it on your phone, and you can recognize right there on the device. And as we'll see later, there are other use cases where it's preferable to have the recognition happen in the cloud. So as this Merlin effort was taking shape, each time any of us on the Visipedia project gave talks about birds, invariably people would say, well, that's cool, but can you also use it for mushrooms or plants or interior design, postage stamps, and so on? And basically, the answer to any of these questions was yes, if you can furnish access to a community that will share images, provide annotations, do focus group testing, and so on. But out of all these different taxonomic groups, the biodiversity community, and to some extent the food and nutrition community, were the best partners because they had abundant data and such a desire to share it and work closely with the machine learning community, essentially without privacy issues, or with privacy issues that were very manageable. So this gave rise to what we call the FGVC workshops, fine-grained visual categorization. And these are now roughly a regular fixture at CVPR. I say roughly because we have to apply for that workshop every year. And knock on wood, we just submitted the proposal for the latest FGVC workshop. But at each of these workshops, we organize competitions, and you can see there are things like camera traps and birds, insects, and so on, as well as food and nutrition. And we also invite guest speakers from these different communities of knowledge. So it's really kind of a matchmaking endeavor, bringing together computer scientists, data science people, computer vision, and so on, experts to discover interesting problems in these different domains. Some of the research problems that have popped up there are, so just to remind you like what fine grain looks like. At the top left, you see two different species of ladybug, and one of them is 2-spotted and the other is 7-spotted. So at a glance, you might not notice that these are different at all. So there's some really subtle field markings there. But in terms of research challenges, one of the things that's really pronounced in the fine grain world is the so-called long tail problem. So in the old days of computer vision, if you did object recognition, you would just collect objects that are sitting around the house or in a lab and just have a uniform distribution over training examples that you have for all of them. And this just isn't what it's like in the real world. The real world in many, many domains, whether it be medical or biodiversity or clothing and apparel, we observe a long tail phenomenon. So what that means is that, for example, with plants and animals, is that there's certain categories like this monarch butterfly for which we have a huge amount of training images. And then this is shown on a log scale, but there are certain species like this type of flower or this type of insect that have very few, maybe even just a handful of examples. So this is what in machine learning we would call a few shot territory, or even one shot if you get down far enough. And this is something that was ignored in computer vision for a while, and now it's just understood that we need to be able to train systems that can handle this long tail and perhaps make use of features that are learned for related categories in this part of the curve, and use that to benefit some of these less well represented entities that are down here. And that's also related to this this notion of domain similarity and pre-training and domain transfer, where as you can kind of get an economy of scale in a sense, where if you're working with many fine-grained categories, and let's say that one of those categories is birds, and you'd like to expand to a new domain like butterflies, it could be that the butterfly and moth community just simply doesn't have that much data available that's labeled. So that would be a form of a long tail problem or few shot problem. But if you qualitatively or quantitatively can determine that a lot of the features that would be useful for identifying moths and butterflies are also useful for birds, you could do a kind of domain comparison and borrow the features from those related domains and use what is now a pretty commonplace concept, which is pre-training a network and then fine-tuning it on your particular target domain. So these efforts with the FGVC workshop, and in particular with the California Academy of Sciences, gave rise to species identification tools in the iNaturalist app. So this is another app that's also free. So those of you that perhaps someone on this call looked up the Merlin app, you can also look up iNaturalist. It's free. This one is cloud-based, so you don't need to download packages. And before Visipedia worked with this team, you would take the picture, make a guess of the genus and species, put it onto the iNaturalist social network, and then wait 1 to 2 days for another person on the network with sufficient expertise for that taxonomic group to confirm it or correct it. And at that point, the record would become what's called research grade. And research grade is what we use to train the models periodically. Now with Visipedia functionality, you take the picture and you can click on a button that says suggest species. And then you get this short list of suggestions that are visually similar and if possible, and if available, seen nearby. And then you can tap on that, again, put it onto the social network. It's not automatic. It's not just put directly into the training bucket for that species. You still have to wait for someone with sufficient expertise in the iNaturalist community to confirm it. So, so that was just a quick overview of Visipedia. And when I talk about GBIF at the end, you'll kind of see how we're aiming to take that global. But I want to describe, before we do that, something that is less in that conventional supervised learning paradigm, something that is a little bit more exploratory in nature, because sometimes you want to solve problems where the category labels are not well known, or you just want to discover something new that that shares some characteristics with things you've seen before, but you may not know what it's called. There may not be a community of expertise behind it. And the kind of thing we're tapping into here is the notion of relative similarity using a triplet formalism. So here's a case where even if you don't know what the species of these birds are, we could look at images A, B, and C, and just using your basic human visual system, you could say, well, image A and image B look pretty similar. They look more similar than either of them look to this image over here. Okay, so that's an example of a triplet-based comparison. It's a very quick, simple relative ordering that doesn't require you to map these images to a semantic label. It's just saying these look more alike than any pair formed with this one. And if you look at relative comparisons in the literature in general, a lot of approaches that involve things like metric learning or collaborative filtering, when you're collecting training data, they're based on looking at pairs. So here's an example of a pair of stimuli. And the question that is pondered by the human observer is, do these two foods taste similar? So in this section of the talk, where we have a statement that we put out, these two foods taste similar. And as you go through the survey, you're indicating your level of agreement. So if you do this as a pairwise task, and you use a Likert scale with 5 or 7 steps, it's tricky because, especially if you come into this cold, you're not sure what this scale means. And as you go through it, you might change your mind about what those different levels mean. So suddenly you're looking at a dessert and something that's not a dessert, and you might change your mind, or there could be some kind of drift in terms of what you think constitutes a level 3 similarity. But again, if you're looking at these relative comparisons, just empirically, we found that it's less of a cognitive burden. It's just easier to go through big batches of these. That said, if you're dealing with triplets, then you're running into a potential cubic complexity problem, which would end up being very boring in its own way for the observers, because if in its most brute force form, if you just presented all possible triplets from a corpus, A, B, and C, and the human observer had to go through all of these and indicate which two were most similar, it's really repetitive. You're doing these nested for loops, and they're going to feel like they've already answered the same question many, many times. So we introduced this idea that we call grid questions, and this was in an HComp paper about 4 years ago. And the idea here is, instead of making use of a triplet with images A, B, and C, what used to be represented by images B and C, we grow into a grid. So it could be a 3x3 grid, or let's say up to a 5x5 grid that fits comfortably on your monitor. Now, what we're doing here is asking the observer to select K out of the N images in that grid. It's an N choose K problem to indicate relative similarity. And in particular, we're tapping into the fact that the human visual system happens to work very well in parallel. So humans have very effective parallel processing when it comes to object recognition. From a machine perspective, it wouldn't make any difference. If it were just a robot answering these questions, there's nothing more to this than the anchor image A, a set of stimuli in the grid, which the machine would still have to loop through and consider all the constituent triplets. But from a human perspective, we found that when we put these tasks on Mechanical Turk, even though we are getting more bang for the buck per screen, so each time we pop up these carrots and this grid, and someone selects, in this case, 4 out of 9, they're indicating that those 4 images are closer to these carrots than the remaining images in that grid. And by doing that, we're able to harvest a large number of triplets where we know that we can form essentially these triangles that will say that these are the ones that are less similar with respect to any of these pairs. Just from a bang for the buck perspective, like payment and job satisfaction, like how much the Turkers enjoyed the task, we got more data and happier workers because they liked looking at screens full of images of food. Now, there is some noise introduced because if this is a forced choice. So here we had a forced choice of 4 images they had to click on out of 9. Well, it's certainly possible that there weren't 4 images in there that had sufficient similarity to these carrots, but that's okay. It actually makes it simpler, and any algorithm that's going to do something like metric learning from this type of data is going to have to handle a certain amount of noise in any case. Now, remember, in the supervised paradigm, what's typically done, for example, if you wanted to analyze images of food, you would take a deep network. Suppose it has 1,000 categories. So in the supervised paradigm, you would have names of all of these dishes. It could be cuisine and dish name. So you would train this essentially like you would for, let's say, ResNet 50 for ImageNet. In this case, it would be for food. You have 1,000 different dishes. You train the network. But remember, this only works for the dishes that you know in advance. So what do you do when you run into problems like you have some familiar food, but it's in a state that you didn't recognize before? Or perhaps you didn't take context into account. So if I just were to focus on this green part, would you know if that's guacamole or wasabi? What if you put a tortilla chip into this thinking it's guacamole and took a bite? You'd get pretty mad at the app for making that mistake. But it's probably the case you can look at this and see how it's presented and get a sense of what that context is. So there's some benefit we would get by having a more exploratory approach to this problem, something that's kind of learning more adaptively. Then you get these strange cases like, I don't know if this is real, but suppose it is, a wasabi Kit Kat bar. That's kind of a genre-defying thing. It's sort of a dessert, but I wouldn't think of wasabi as a dessert kind of thing. Where does that fit in your hierarchy? Chances are you didn't have a node for that, but it would still be nice to be able to put that into some kind of high-dimensional space and make sense of it. There are also things where there are different cultures that give rise to culinary traditions, and they produce very similar foods, and they're called different things. And so if you're in a new environment looking for something to eat and you're overwhelmed by all the terminology, perhaps you just want to look at pictures and you're not too concerned about what it's called. It's more of a visually driven menu concept. So this is just some speculation here, but what this brings us to is what we call snack. And it's purely coincidental that it ended up being snack. We were working on food, maybe subliminally that influenced the name of the algorithm, but it stands for Stochastic Neighbor and Crowd Kernel. And so first, if I were in front of an audience right now, I would ask actually there's a hands up feature. So if people want to try that, they can. How many of you have heard of T-SNE? Okay, I see one hand. Okay, so T-SNE, also known as TISNE, is a tech. Oh, I saw another hand go up. It stands for Student T Distributed Stochastic Neighbor Embedding, and it's indicated through this pipeline on the bottom. T-SNE is used for low-dimensional visualizations of high-dimensional data. So what you would do, for example, is take that conventional deep learning pipeline, like the one I showed for recognizing food. You peel off the final layer, so the one hot encoding softmax layer, you just get rid of it. And in this case, you would have a raw 1,000-dimensional vector. So you put in an image of food over here, you get out a raw 1,000-dimensional vector. So every food image in your data set would get mapped to that very high dimensional vector. So that's pretty hard to visualize on its own. So T-SNE proceeds in the tradition of multidimensional scaling by considering, in principle, the pairwise distance between all of those 1,000-dimensional vectors. So you put in maybe 10,000 images of food over here, you get out that many instances of this high-dimensional vector, set up a dissimilarity matrix, feed it into the T-SNE algorithm, and get out a two-dimensional embedding. So ignore this pathway at the top for now. You would get that high-dimensional data, look at the dissimilarity matrix, run it through T-SNE, and then get out this two-dimensional embedding. So with that two-dimensional embedding, you can use color coding to indicate the classes. And whether it's MNIST digits, SciFAR categories, it doesn't matter. You can get this two-dimensional representation. And what you're hoping for is that points that are nearby in this 2D embedding would also be perceptually similar in the input pixel space. So in a task like trying to predict whether two foods would taste similar, it's possible that this conventional pipeline at the bottom here would solve it for you. So you could do this empirically, look at this mapping, select pairs of points here, and then focus group test it. Just bring in volunteers with lots of snacks and have them determine whether that works. What we're doing with the snack algorithm is adding something to this cost function based on human expertise. So that pathway is represented by something called T-STE, which is Student T Distributed Stochastic Triplet Embedding. So think of it as the sibling of T-SNE that works on triplet-based data. So if you've collected data the way I described it, and you don't need to do it for all triples, you can do it for just 0.1% of the data. So you give those grid stimuli to human observers, have them click on all of those and choose K problems. And then you set up this weighted cost function that's partly T-SNE plus some constant times T-STE. And we tune that on some representative data sets. And what we get is this concept embedding where it's essentially the T-SNE pipeline, except the embedding has been pulled on by the human preferences. And that constant in the cost function allows you to determine how much that human expertise should influence that embedding. So if you apply this to something like Food 10K, a database of appetizers, then again, without any notion of semantics at all, you can get out this two-dimensional menu, visual menu, where you have berry-based desserts over here, waffle-type desserts here. Some of these have nuts. And so you can see like chocolate-type things here. So it's the kind of thing where you can roam around this space of food and get a sense for what things, at least a representative group of observers would say taste similar. More quantitatively, you could use a system like this for quality assurance. So suppose you had a very elite, high-priced set of experts that are annotating bird data for you. So you take a large data set of 200 species of birds, have experts annotate them, and then you just want to see if perhaps they made some mistakes. So what you could do is take that data set of 200 species of birds, throw away all the labels. So just pretend that it's an unsupervised problem. You just have images of birds. And then set up those grid stimuli and put it on Mechanical Turk or some crowd work platform, and just ask people to indicate which birds look similar, which ones look dissimilar, again, without any notion of what the birds are called. Okay, so run it through Snack, produce the embedding, which is shown here, and then put back the labels. So show the labels in terms of this color coding, and then look for anomalies. Okay, so and in particular, in this case, when we did this plot, we looked at this example and saw there's a blue dot here that seems to be surrounded by some green dots. And we went back to the Cornell Lab of Ornithology and asked them, do you happen to know this person? So we clicked on it, we saw which expert labeled that example. And they did know who that was. And we said, would you mind asking them about this particular woodpecker? And they did, and it turned out it was a mistake. So the person that labeled it just clicked on the wrong entry in a pull down menu. And they said, yeah, I got that wrong. So that needs to be fixed. And so they fixed it in the next round. And so the point here was that was detected by a crowd that didn't know the name of the species. They were just observing basic visual similarity. So you could imagine the same kind of thing happening with tissue pathology or postage stamps or all sorts of different visual stimuli, again, just separate from that specialized knowledge of what things are called. It's essentially a way of doing QA with an audience with general visual perception capabilities. Okay, so now let's go back to the original thread of what you saw that led to Merlin bird photo ID and then iNaturalist. Where does that progression go? So how do we take that kind of Visipedia powered app and scale it at a global level? Well, it turns out the right group of people to talk to about that is GBIF, the Global Biodiversity Information Facility, and they're located in the University of Copenhagen. And so after working with enough biodiversity researchers, we started seeing the term GBIF all over, and we just finally asked them, what is GBIF? And so it's a global aggregator of biodiversity data. The Cornell Lab of Ornithology happens to be the biggest contributor to it. iNaturalist is possibly the second largest contributor. And there's thousands of other citizen science groups, museums, herbaria, and so on, that have their own research projects, citizen science efforts, and so forth. They may be running their own operation anywhere around the world, but on a weekly or monthly basis, they push all of their occurrence data to GBIF. So the GBIF headquarters are in Copenhagen, but they have global representation. I think around 50 different countries have nodes of GBIF. And so they mirror all this data. And essentially it's a one-stop shop. If you want, for example, if you're a property developer in northern Sweden and you want to make a large office building, you could select a rectangle around a plot of land, go to GBIF, pull up all the species that have been observed there, and find out if there are any endangered species that need to be taken into account, right? So you wouldn't need to do that for hundreds of different stakeholder museums or citizen science efforts. You just go to GBIF to do that, and then you find the pointers back to the relevant multimedia and metadata. So this just gives a sense of the scale of the operation. And so when we talked to GBIF, we said, you know, we really enjoyed working with the Cornell Lab of O to make Merlin and California Academy of Sciences for the iNaturalist app, but our small academic team can't scale like that. We can't send one of our PhD students to all these different institutions that would like this kind of functionality. So let's work together in some way and figure out how to take this tech stack and expertise and bring it to lots of different communities. So GBIF was very interested in doing this. The main team at GBIF they focus on database techniques. So they're not AI experts per se, but they're very aware of what AI can do. And so they volunteered essentially to serve as mediators between the AI community and the biodiversity community. So because so much of the biodiversity community already has their data shipped to GBIF on a regular basis, in principle, we can work with them, Visipedia and GBIF can work together to flip a switch that just brings AI functionality to the data that's already there on GBIF. Of course, it needs packaging, it needs proper treatment of licensing, citation, and so forth. But these are things that GBIF knows how to do just from their role as a database management operation. So we worked with them to do this. And just to point out, this isn't just images that are captured in the wild. There's herbarium sheets, historical drawings of species, x-rays, microscopy images. So there's pretty diverse images in GBIF. And so we then produced what we call this mediated machine vision cycle. And essentially it's a publishing operation. And it's facilitated by Google Research and the TensorFlow Hub group, which is part of Google based in Zurich. So the idea here is suppose there's a group that studies mushrooms and they would like to make their own mushroom app for some region. Now, that group, that hypothetical group already sends their data to GBIF. So they let GBIF know, we would like to make an app that has image recognition functionality. So GBIF, can you package a data set for us? And so GBIF says, yes, we're delighted to do that. We'll package a data set with 1,000 species of fungi, and we'll give it a DOI, a digital object identifier. And so in the biodiversity community, and to some extent in machine learning, but the biodiversity community uses DOIs for everything. So you can use them for citation, attribution tracking, but essentially, normally you see DOIs for publications, but it can be for anything. So GBIF talks with that mushroom group, packages a data set, gives it a DOI, then shares that data set through the GBIF API with Visipedia and or Google Research engineers, who then select a suitable architecture, pre-trained most likely, fine-tune it for that domain, train a model, give the model a DOI, which again GBIF handles. So it's not just the data set, but the model that's trained also gets a DOI, hosts that on TensorFlow Hub. So in this case, the group that wants to make the mushroom recognition app would become a publisher on TensorFlow Hub. And then that is released on TensorFlow so that if any third party, Android or iOS developer, wants to make an app that can recognize mushrooms, they can go to TensorFlow Hub and they can preview the model. So they can pull up the page for it and drag and drop sample images without knowing about details of machine learning or neural nets. They can just try it out, the latest version, in this case, of the mushroom recognizer. And if they want to use it, it's freely downloadable. They just need to cite it. So in their app, they'll cite the DOI for the data set and the model. And this is great for the people who publish the model because they get citations, they're able to write grant proposals, indicate the impact that they're having in the real world. And so just closing the loop, this particular group called Svampe Atlas, or the Danish Fungal Atlas, put together an app that on the surface looks pretty much like what we did for Merlin Bird Photo ID or iNaturalist, But the important difference here is that they did it on their own using these tools that were made available through this mediated infrastructure. Okay, so this Svampe Atlas app, which also is available on the App Store and Play Store, this represents the kind of thing that you can do with this new framework. If you look on the TensorFlow Hub page, you can see the so-called provenance graph. So as I indicated, there are many models that were pre-trained on other data sets. And I think it's important to give proper credit to everyone in that chain. So for example, what you would see on a typical page for a publisher would be, we used Inception v4 that was pre-trained on iNaturalist, like a very large general biodiversity data set, with the specific training and validation images provided by this particular competition data set and so on. So you get to see everything that went into it. So, and that's all available to you through that DOI. So that's basically hot off the presses. So that's the first end-to-end example of engaging with the biodiversity community using this mediated machine vision pipeline. If people are interested in that, we have these web pages that indicate how to tap into that. And that concludes the talk. I just want to thank my collaborators from Visipedia at Cornell and Caltech. One of the key collaborators in the FGDC workshop, Ryan Farrell at BYU. Tim Robertson is the director of informatics at GBIF. And our closest collaborator at Google Research, Christine Kaiser-Chen. And most of the Visipedia research over the last 6 years or so was funded through Google-focused research awards. And again, thank you for your attention.
Thank you.